I'll start my talk today with a short introduction in two domains where I'm active scientifically. The first is radiative transfer modeling and how we use it for scaling. And the second part will be then a combination with machine learning for the purpose of plant functional traits retrieval from optical data. And I will really focus on spectral images of high spatial resolution. After a brief introduction, I will then give you two examples, two case studies where we applied basically two different scaling schemes. First one is from Antarctica, drone-based hyperspectral mapping of the Antarctic moss stress indicators. And the other one is in Australian eucalypt forest, again, retrieval of the leaf biochemical traits of these at the leaf level of eucalypts from drone hyperspectral data. Now you see that these case studies have nothing to do with agriculture, and the PhenoRob is all about agro-ecosystems. That's basically because my past research mainly was focusing on natural ecosystems, but what I will be showing today is applicable and transferable to any type of ecosystem, including the agriculture field. So I believe that what I will show you today will be useful to all PhenoRob members. So let me start with definitions of scales in optical remote sensing. Most of you, or many of you, especially if you are a PhD student, are challenged with measuring the plant traits at different scale. And we're talking about temporal scale as well as spatial scale. As we go to the more detailed scale, so high resolution as well, the temporal scales are usually shorter, and then approaching larger scales, we usually acquire data within coarser spatial resolution when it comes to spatial, but as well longer time periods. Now, the desire is usually to acquire data at scales of airborne or satellite observations, because that allows us to get many samples or our fields measured in a relatively short time and have a statistically representative measurements, number of samples, and so on. Nevertheless, we can't understand those measurements taken from aircrafts or satellites without performing or conducting some observations at the lower scales. Specifically, if we talk about vegetation, the basic scale where many people start is leaf. And then going up, we have individual plants, individual stands or fields, ecosystems, and biomes. Now, the leaf is kind of a rational, logical starting point, but when we talk about optical remote sensing, the signal which we measure is actually formed inside of the leaves. And so we go even further down to tissues or individual cells or even cell organelles scales, spatial scales, because that's where the particle of light, which we use in optical spectral remote sensing as the carriers on the information, are interacting, are having active exchange of energy between the objects they actually can interact with. So the problem with remote sensing in general is we are in need of moving from these interactions taking place inside of the leaves all the way to top of canopy or even above the atmosphere, within the atmosphere, taken observations. And this scaling, we usually call upscaling, is providing us with additional hurdles and complications. It imposes on us a little bit higher complexity in our data, and it has to be somehow tackled scientifically. So let's first talk a little bit. Let's start from top down, from our remotely sensed data. Many of PhenoRob scientists are using drones as an acquisition platform. There is a lot of RGB imagery being processed or multispectral images with the high spatial resolution. Now, the problem in remote sensing of the canopies is anisotropy of the signal which we are measuring at the drone level. What do I mean by anisotropy? I mean angular anisotropy, which is mainly driven by the structure of the canopies we are observing. So if we measure or take our images, they usually measure light reflected from the objects. In our case, it will be canopies composed by leaves, stems, and as well the background, which would be, in case of agriculture field, bare soil. And the pixels, depending on the size, so on the ground sampling distance of those image pixels, will contain different type of information. Of course, if we are acquiring data with a millimeter resolution, we can distinguish individual leaves, and then we can separate those leaves and work with them as a unique object. Nevertheless, that's not always possible. If we work with hyperspectral sensors on the drones, the highest resolution we can work with will be around 5 to 10 centimeters. In that case, our pixel is already containing as well multiple number of leaves or some background, shadows, and so on. This complex composition is basically causing this angular anisotropy of the signal of the reflected light, which is depicted or described on this graphic here. So if we take any band of any reflectance, but let's say in most prominent structural impact is in the near infrared bands, and then we observe the reflectance from different viewing angles with respect to the solar illumination angle, then within the solar principal plane, which is the plane between the sun and our sensor, we will obtain different intensities of reflected light. And the function will look roughly like this. It will have a very steep peak, which is something what we call hotspot, and then it will have very deep trough here, which is something place which we order geometrical arrangement, which we call dark spot observation. Dark spot gives us the lowest reflectance, hotspot gives us the highest reflectance. Now, how these are being shaped up, basically, it's the combination of how many facets or the objects are illuminated directly, what is the ratio between shaded and sunlit parts of the canopy. And of course, there is as well a component of the solar zenith and azimuth, mainly the zenith, which the sun at the highest zenith angle will provide us with more energy and as well possibly more sunlit leaves. So the hotspot is very hard to measure. We can measure it with goniometers or we can measure it from airplanes, but you see the problem is that we basically are in the solar illumination angle, and so we can see our own shadow. That's why it's very hard to measure, but that's the signal which has the most light. Now, most of the measurements we do in remote sensing is from the nadir, so it's the perpendicular straight down measurement. And you see that the reflectance intensity signal is somewhere in between the hotspot and the dark spot. Now, depending on what will be the solar position, this intensity will as well change. So that means it's very hard to use the intensity of reflected light directly as an indicator of any internal composition of the leaf, biochemical composition, or even the structure, because it depends on this three-dimensional distribution or arrangement between sun, sensor, and our ground. So what are the main reasons of the plant canopy reflectance anisotropy? First of all, it will be foliage distribution, means what is the leaf area index or density, what is the clumping of the leaves within the canopy. Then the next one is the foliage geometry, the leaf angularity, very often described by leaf angle, average leaf angle, or the functions of leaf angles. Then the next is spatial distribution of the plants within the field. So what is the canopy closure? And then a propagation of shadows. Complementary to that, we as well have to take into account that there is a roughness of the terrain, even though many fields are rather flat, but they can be on a slope, having certain exposition, and that as well impacts the anisotropy. And finally, there are not only plants. Sometimes in the field you can have debris, you can have a litter. There can be as well exposed bare soil. So influence of the other canopy elements and ground surfaces is part of all that. Now, how do we deal with all these confounding factors if we want to retrieve information which comes from the leaf, from inside of the leaf, but these factors are part of our signal? We need to basically either embrace them or try to reduce their impact. Now, the scaling or spectral spatial scaling in case of the optical remote sensing is about the techniques which are dealing with exactly this phenomena. We have two types of the scaling, let's say, traditional one, and the first which has been applied was a direct empirical scaling using the lab field measurements, where we measure the traits in the field, and then we directly link it with the optical remotely sensed observations. Now, this is very easy and fast and effective, but it only works at the local scale. It can be inaccurate to a certain extent, and it's site-specific. And possibly as well, if we just try to fit the relationship between what we measure at the ground and our remote sensed signal, we might miss the causality, so the cause effect might not be established as a primary cause effect, but we can be looking into secondary or even tertiary effects. So the other possibility is actually to embrace this complexity of geometry and plug in between our leaf measurements and our canopy measurements physical models, which can mechanistically simulate all these, let's say, confounding factors. And we can include these simulations in our interpretation algorithms. These physical models we call radiative transfer models because they mimic transfer of light between the sun and the object, which is canopy or leaves, and then the object and the sensor. They include usually interaction in atmosphere as well as on the lens surface. They are of mechanistic physical nature, and that's why they are transferable across sites and spatial scales to a certain extent. The cons of these approaches, they need long development time. They are rather complex. We need to understand the physics behind those interactions, and they, of course, therefore, require more inputs and intensive computation. Nevertheless, in recent times, they can be effectively combined with machine learning which provides us for today, let's say, the middle way, the optimal way, so the combination of these physical and statistical approaches. Now, the radiative transfer models are around already for several years. I would say the development started with atmospheric radiative transfer modeling already in 70s of last century. And then from 90s of last century, we could see development of first one-dimensional models at the both scale. So there are models which are mimicking the interactions of the light within the leaf structures, as well as inside of the canopies. The one-dimensional model means that the only variability which we are simulating is in a vertical dimension. Horizontally, we assume the medium, being leaf or canopy, is completely homogeneous. So there is no, let's say, structurally or interruption, discontinued canopies. There is only one large canopy. So this can be used in agriculture very well for some simple canopies like wheat, barley, or any grasses and so on. Now, of course, for more complex canopies like savanna or boreal forests, we have to consider as well discontinuity in horizontal direction, and therefore three-dimensional models which can embrace the complexity in all three spatial dimensions has been developed. We have again three-dimensional leaf-level radiative transfer models and as well the three-dimensional canopy models. If you want to know a little bit more about it, please follow the DOIs at the bottom of the slides. Usually they are providing more detailed information. Now at the leaf level, the current working horse of radiative transfer models is ProSpect Pro or FluSpect CX. These are two most commonly used 1D semi-empirical models. These models are approximating the leaf structure, which looks like this here. This is the three-dimensional expression simplified of the leaf as a pile of layers. The layers are assumed to be cellular layers, which are separated by air space as in between. And then the model is using 2 or 3 basic laws for radiative transfer simulations. It's a reflection and refraction law, which is simulating the scattering of the photons inside of the leaf, and then the exponential extinction Lambert-Beer law, which is driven by absorbers inside of the leaf. And this particular model has as absorbers chlorophyll content, carotenoid content, anthocyanin content, water thickness, and dry matter content, which was recently split in nitrogen-based proteins and carbon-based constituents. So using these few inputs, the model is able to mimic the leaf reflectance, leaf transmittance, and if we subtract the sum of these two from 1, we will get the energy which has been absorbed in between 400 to 2,500 nanometers. So this is what we call optical part of electromagnetic spectra. So this is the first scaling step where the photons, which due to their size and the wavelength basically interact inside with the leaf constituents, can be transported, or their these processes of scattering and absorption can be expressed as a fluxes of leaf reflectance and transmittance. The second step, which is needed, is actually taken at a canopy level. And for the canopy level, we have, again, as I already mentioned, one-dimensional or three-dimensional solutions, which can be either programmed for stochastic representation of the canopy. So that would be something what we called homogeneous turbid medium, or deterministic representation of the canopy, where we have the actual objects, actual leaves represented by geometrical primitives like triangles. So it can be meshed individual plants. Now, the solutions for radiative transfer, the first ones and the earliest ones were analytic turbid medium 2, 3, or 4 flux solutions based on Kubilka-Munk theory. These solutions assume, as I said, one-dimensional canopy built by infinitely small leaves, and there are quite a number of approximations for multiple scattering and hotspot modeling. So the models are not very accurate, but they are very fast and very efficient. When it comes to 3D canopies, we have far more solutions. There is kernel-driven solutions, which are working with volumetric and geometric scattering and account for the shadows, including the hotspot. Then we have geometrical optical models, where the canopy is described by volumetric shapes, like here, this spherical objects, for instance. They can therefore simulate horizontally discontinuous canopies. Then we have flux tracking in the discrete geometrical models. These models can deal with n fluxes in an iterative fashion, simulate the multiple scattering, and their canopies are represented by voxels, so three-dimensional pixels filled with the turbid medium of leaves. Finally, the most recent and most accurate are Monte Carlo photon ray or path tracing models. These models are resolving every single photon object interaction, and they can simulate very exact plant shapes built out of these optical geometrical primitives. There is one new solution which is called spectral invariance. You can stumble over it in the publications. It is not per se radiative transfer model, but it's a theory based on photon recollision probability. Again, more information can be found in the literature. I will move more towards how we can use these models for the scaling, or how we basically can retrieve the quantitative parameters of vegetation, either biophysical or biochemical, from optical remote sensing data. So the first way how we can do it, and this approach relates to this first empirical scaling, is just establishing parametric regression between our observation in a field and from drone or airplane or satellite. We can as well establish non-parametric regression using some machine learning algorithms like partial least square regression, random forest, neural networks, etc. These are, as I said, straightforward without any physically based scaling. The other option is to engage radiative transfer model and invert the model itself. So that would be a mathematical inversion where we couple the leaf and canopy radiative transfer model and then establish through some cost function and try to find the ideal solution or the optimize basically solution for our remotely sensed observation. Finally, the last type which we used mostly in remote sensing, optical remote sensing, is a combination of the forward radiative transfer modeling and non-parametric regression, where the forward RTMs are producing so-called labeled data, and then we train on those labeled lookup tables, so database of canopy reflectance signatures simulated for many potential scenarios. The non-parametric function or machine learning algorithm, let's say. So these are the 4 types, and I will today present 2 case studies for empirical non-parametric regression and hybrid retrieval. Now, please don't mix up the hybrid retrieval with the hybrid machine learning. These are 2 different things. What I'm talking about is a hybrid retrieval using physical modeling and machine learning. So my first study case is drone-based mapping of Antarctic moss stress. Now, why we are interested in this problem? The Antarctic shore is populated, not everywhere, but wherever there is fresh water, liquid fresh water available, by mosses. These are the only higher plant forms growing in Antarctica, in true Antarctic environment. But in past few decades, last 20 years, there is a steep descent, or the steep, let's say, stress impact on those moss beds, which are then turning in so-called moribund status, dormant status, and they don't provide all the expected ecosystem functions for the Antarctic ecosystems. The reasons can be twofold. One is high UV radiation. You probably heard about the ozone hole above the Antarctica, so that's one of the causes. And the other cause is sunbaking. When we are getting more clear days in the summer season, and the mosses are basically overheated. Now, before we do any remote sensing of this phenomena, so mapping these stresses, we would like to understand how we should approach it and what are the best indicators of those stresses. And then we have to go back to physiology of the moss and somehow learn how they react to the stresses. Now, if the environment is in optimal, and that's very rarely, but if the moss gets all the optimal growing conditions, which is fresh water and just a right amount of the irradiation, it will flourish, it will basically produce leaflets, and it will accumulate a lot of photosynthetically active pigments, mainly chlorophylls. Then if the water, fresh water, disappears because it is just basically evaporated or there is no enough snow melt, then the drought will cause, as a first reaction, a rolling of the leaves. So you see on this cartoon here, basically the leaves will literally roll, and this phenomenon is not only known for mosses, but it's being used as well by other plants like maize or sugar beet, for instance. And this is taking, the time lead for this is hours or few maximum days. So this is the first reaction on the drought. Now, if the sun increases the intensity, then we talk about the sunbaking. And in that case, after a few days of sunbaking, the moss will lose the chlorophyll, will basically destroy the chlorophyll, will discolorate to rather red or brown colors. And so we have a decrease of the chlorophyll content as a second indicator of the longer stress. We can look at these stresses in laboratory as well and simulate them. That's what we did. So before we did any remote sensing, we moved some of the moss patches into the lab and we exposed them to different stress scenarios when we were not irrigating, and using higher lights, to see how these stresses can be observed spectrally. So we measured those moss patches every few days with the handheld spectroradiometer and produced hyperspectral signature of stressed and unstressed moss. This is example of one of our results. So when we have moss, which was originally taken from the environment, this is our time t0. We expose it to the stress. means we didn't give it any water for a couple of days. And then you see that in the near infrared reflectance, this is a signature of moss canopy reflectance, we have a significant and obvious decrease of reflectance. That's because the rolling of the leaves and then the photons being able to penetrate deeper in the canopy and eventually being absorbed down there in lower layers. Now, if we give it again water back, it will basically spread out the leaves and then make increase in the near infrared. And as well, it will produce more chlorophyll. So you see there is a deeper absorption in the red bands and higher reflection in the green bands of the visible electromagnetic spectrum. Now we can use this knowledge to build our interpretation algorithm for drone observations. What we did, we basically took all the measurements, and we had hundreds of these different measurements, and we trained support-vector regression machine by putting as an input data a reflectance of a given wavelength, where we knew the change is triggered by these specific things, like chlorophyll content and the leaf rolling, so leaf density increase. And that was our target or the output for the training. Now, the training was pretty successful for chlorophyll content prediction. We got quite a good relationship with systematic error being much smaller than unsystematic root mean square error, which indicates the model is pretty stable. Most of the error comes from the noise in the data. The same for the leaf density. Now we talk about effective leaf density, which means how many leaves in 1 millimeter of shoot we have, including the rolling. So we again achieved quite acceptable RMSE. So we trained this purely on the laboratory measurements. And then we flew over 2 moss beds in the eastern Antarctica, close to the Casey Polar Station from Australian Antarctic Division. We acquired V-near hyperspectral data of 162 bands, and basically then applied our support-vector regression per pixel on the moss canopy. Now, first we had to separate the moss from the rest, which is the background, which is not of our interest. And as well, we separated moss, which was photosynthetically active and inactive. So the one which was dead, we didn't really analyze. And then when we applied per pixel this SVR, we could estimate chlorophyll content in nanomoles per ground dry weight, grams of dried weight, and as well the efficient leaf density, which is how many leaves in 1 millimeter length of the shoot we have. And these are the quantitative maps, which could be actually combined in a final map, which is a relative Weiger. Just to point out, basically the lower density, the higher health, and the higher chlorophyll content, the higher health. So these two are negatively related. And then we combine them in a relative Weiger, we call it, which is somehow easier to interpret. And you see the parts of the moss beds which were pretty healthy, the blue parts, and which were quite stressed are the warm colors. When we overlaid over this map the melted water runoff streams, we could see that really the healthy parts are sitting on the basically discharged streams from that area. We did as well compared our results with satellite observations to see if there is a potential to use satellite data to do that. This is the same retrievals. We found out that for leaf density, the satellite is a little bit overestimating, while for the chlorophyll content is underestimated. We used WorldView 2 multispectral data. So in other words, the satellite would basically make the assessment of the moss health worse than it really was. Okay, so that was the first scaling, which was simpler, straightforward, could be done because the canopy is simple and it's rather homogeneous. So, but for more complex canopies, we really need to plug in that radiative transfer modeling, and that will be done, or we did that for the forest in Australia, one forest study site. The approach for retrieving the functional traits here, as I said, is combined physical and machine learning. So we always have to start with ground measurements. Then these ground measurements were used to parameterize structurally the forest site, as well as from the biochemical point of view. Then the radiative transfer models are doing for us the scaling, as I said, to produce a virtual database of simulated canopy reflectance images. And these are used for machine learning to train the retrieval algorithm, which is then applied on a real drone or airborne or satellite data reflectance images per pixel to obtain the map of quantitative vegetation trait. In this case, we have a chlorophyll content map per pixel of the image for sunlit parts of the grounds. And this can be validated if we have any measurements. Now, this scheme we called upscaling scheme, and can be basically used generally for any other ecosystem. The radiative transfer model, which we used here for this case study, is called DART, Discrete Anisotropic Radiative Transfer, is based on the LuxCore render library, which is the open source rendering, and it was repurposed to simulate spectral radiative transfer by bidirectional Monte Carlo vectorized path tracing. The unique thing of this radiative transfer approach is that it's only tracing photons ending up in our sensor. So if we have a sensor, we basically first probe which photons go to the sensor. We then trace them backwards to the source, which is in our case sun. And we as well use interconnection between these path lengths to compute the scale basically, or simulate the interactions of the light within the canopy and as well in atmosphere. As a result, we can get images like this. This is a false call composite near infrared image of eucalypt tree from nadir with very, very high spatial resolution. Now, of course, there are some inputs which we need for parameterizing that model. So first we have to set what canopy we are going to simulate. In our case, it's a tall eucalypt stand in a Tambaramba supersite. It's one of the permanent monitoring sites in Australia where we know the species. There are two dominant species of eucalypts, and we know every single tree position, which was advantage as well for creating a three-dimensional representation of this site. How we created the trees, which we need for scaling their structure. So we need to basically put the trunks, we need to put the leaves, the right leaf area index, the right leaf angularity. That's how we include that structural component. We used terrestrial laser scanning. We scanned few trees, which then their point clouds was split between the foliage and the wooden points. So the points means the echoes from the LiDAR, and then we used a specific AddQSM algorithm to reconstruct the wooden components, and we used the leaf point cloud as an attractor for distributing leaves. So the distribution of the leaves was done per vertical horizontal layers of canopy, where the density of points was actually taken as attractors, and then we had these prototypes of the leaves or the actual leaf shapes made in a blender, which we distributed through the canopy based on the point cloud density. Now, of course, we couldn't reconstruct every single tree of the site. We only reconstructed 19 representative trees. which were selected based on the tree height and DBH, so diameter of the trunk at the breast height. And then the other trees, which were other 402 trees, were actually represented by scaling those 19 representants. We picked them up based on the height and then rotated and scaled based on the knowledge of the tree height. So that's how we got to all the representative trees. We as well try to compensate for the occlusion of TLS point clouds, so we don't see every single leaf due to the shading of the leaves. We have a special algorithm developed where every single of these 19 representative trees was corrected for the occlusion. Now, this is pretty long process, as you see, but at the end, we ended up with creation of the 3D representation of that forest. I'm sorry for my slides. They are approaching a little bit faster than I want. So this is the final 3D mock-up, we call it, or the representation with all the trees and with as well understory, which are shrubs and grass, which were taken from as well TLS data and reconstructed using AMapVox Python library. So we ended up with the actual representation of the forest, which was flown with the hyperspectral sensor head-wall micro hyperspec in September 2019, and we basically together with this overfly did some leaf collection for referencing and measured the leaf optical properties and as well biochemical composition in terms of chlorophyll, carotenoids, anthocyanins, dry matter, water content, and use this to verify and recalibrate the leaf level model called FluSpec CX. So we use both the reflectance transmittance measurements as well as wet biochemistry results to make sure that we can, in the DART, initiate the leaf radiative transfer correctly with the FluSpec CX. Now, finally, we are getting to the results where this first slide shows the actual simulation on left-hand side and the actual acquisition of the top of canopy on right-hand side. You see that we have a certain structural reassemblance. The difference is only in color because the trees in DART simulations had the same optical property, all the trees the same, therefore they are not differentiated in RGB composition. Now, but if we take the hyperspectral signature of the entire site and compare it between the DART simulation and the drone observation, we end up with quite good similarity. We have very close overlay between simulated and measured data. And finally, we took only sunlit parts of the crowns because the trees are the object of interest for us. And then we used the FluSpec combination with the DART model to simulate 2,268 reflectance images, where we varied the chlorophyll, anthocyanin, and carotenoid content in the leaf. And we trained random forest regression using this simulated DART-like simulated data in order and applied per pixel to retrieve the chlorophyll, carotenoid, and anthocyanin contents. So again, these are quantitative contents per pixel, which at the end can tell us where we can, what is the pigment pool of individual trees and how big is the variability of the pigment pool inside of individual canopies. So you see here the combination RGB where the individual pigments are prevailing. We have either red color for prevailing anthocyanin, green color for prevailing chlorophylls, and blue for prevailing carotenoids. Now we as well try to compare the results with retrieval from satellite and the laboratory measurements. The ballpark intervals which we retrieved agreed more or less, but when we compare the distributions, which are density normalized distributions of our retrievals, we have some discrepancies, mainly because the laboratory observations or measurements were done on only 8 trees, and we sampled mainly the lower parts of canopy, of course. And in case of Sentinel-2 retrievals, so satellite data and the drone data, the discrepancy is caused by the fact that we have not many shadows in our retrievals. So it is a problem with building up the proper lookup tables. But in principle, we were pretty satisfied, and these maps can be now used further on by biologists for understanding the stress responses of the forest. This is all from my side. I would like to conclude with just 3 simple take-home messages. First message, physically based radiative transfer provides learning data for machine learning algorithms to retrieve plant functional traits from spectral remote sensing observations. Speaking of first case, the hyperspectral drone-based technology and machine learning were found to be optimal solution for mapping stress indicators of Antarctic mosses. And finally, the 3-dimensional reconstruction of the forest stand, which by the way can be done as well for agriculture crops much simpler way, allowed for quantitative retrievals of foliar pigment contents from 5 centimeter pixel size hyperspectral images of individual trees. So these are very high spatial resolution data. With this, I would like to acknowledge my collaborators on all that work. Of course, I would never be able to do it alone. So my colleagues from Australia, University of Tasmania, and Wollongong University, CSIRO, as well as colleague from Europe, from Czech Globe Institute, CESBio Laboratory in France. Thank you very much for attention. I'm ready for questions.